Uh, Good morning, students. Today I'm going to tell you something about cell and its organelles, and this is for our paper Cell and Molecular Biology, code BBBM3T100, and I will be telling you about cell and its organelles. Okay, let us come to that. As we know, cell is the smallest unit that is capable of performing life functions, or we can say cell is the structural and functional unit of life. Now, cell has got different size and shape, and we can see this slide shows different types of cells. Poliovirus has the diameter 30 nanometer. HIV/AIDS virus has the diameter 100 nanometer. Similarly, T4 bacteriophage has diameter of 225 nanometer. Red blood cells have the diameter 7 to 8 micrometer. Similarly, a neuron is having the size diameter of 70 micro. Then this is the plant cell, and the diameter of the plant cell is around 10 to 100 microns. And at the same time, if we look at the size of the hen's egg, which is also a single cell, and its diameter is in millimeter, it is 65 millimeter. So cell exists in different size and shapes. Now there are two types of cells, prokaryotic and eukaryotic cells. Prokaryotic cells are the primitive type of cells, and eukaryotic cells are present in the higher organisms. So this is the structure of the prokaryotic cells, and this is eukaryotic cells. Prokaryotic cells have, we can see this cell, this is flagella, plasma membrane, this is cytoplasm, and nucleus. Nucleus, the genetic material or DNA, is not encircled, or it is not surrounded by a membrane or nuclear membrane. So they don't have any limiting membrane. And this is the structure of the prokaryotic cells. And let us come to the eukaryotic cells, or the cells present in the higher organism. And this figure shows the structure of the eukaryotic cells. This is the cell membrane, nucleus, this is nucleolus, and this is the plasma membrane, or we can say cell membrane. Now this structure is an endoplasmic reticulum, and this is Golgi apparatus. This is nucleolus, and this whole thing is nuclear. So different well-defined cell organelles are present in the eukaryotic cells, and we will be discussing about that in details. Now let us first discuss about the structure of the prokaryotic cell. This is the prokaryotic cells. They do not have structures, or they are not surrounded by the membrane. All the cell organelles are just without the limiting membranes. They are just like that, spread and present in the cytoplasm. They are not bounded by any cell membrane, and they have very few internal structure. And they are present in, the example is in all the one-celled organism or bacteria, virus, all these are prokaryotic cells. Now, eukaryotic cells. This is a plant cell and this is an animal cell. They contain organelles surrounded by the membranes. or they are present in most, or the second point is they are present in most of the living organism, or most of the living organisms are made up of eukaryotic cells. This is a plant cell and this is an animal cell. Plant cell, we can see that this is the plasma membrane, which is surrounded by the cell wall, which is slightly hardened, and it protects the plasma membrane. And then comes, this is the nucleus, and inside the nucleus is present nucleolus. This is a big vacuole present. In plants, a big vacuoles are also present. This is mitochondria. This is Golgi, and these are the chloroplasts. Chloroplasts are present in the plant cell. And we know that photosynthesis takes place in chloroplast. Now let us come to animal cell. This is the animal cell. Animal cell has, now this is the flagella, or flagella which can beat or which can move. And now this is the lysosome, Golgi apparatus. This is mitochondria. Ribosomes, they are present on endoplasmic reticulum. This is endoplasmic reticulum. This is centriole, which arises during the cell division, and these are cytoskeletons present on the cell, present inside the cell. Now let us come to the next slide. Now, difference between prokaryotic and eukaryotic cells. Prokaryotic cells are having single membrane, but eukaryotic cells are all bilayered, or the membrane is bilayered in the case of eukaryotic membranes. No well-defined nucleus is present in the prokaryotic cells, or the genetic material is not bounded by any membrane. It is present just like that in the cytoplasm. But in the case of eukaryotic cells, there is a well-defined nuclear membrane, and inside that are present nucleus, which has got DNA and histones. Now, no membrane-bounded organelles present in the prokaryotic cells. And all the organelles are unbound. They are not bound by the membrane. But here in the case of eukaryotic cells, all the cell organelles are membrane bound. Now let us come to lysosome. Lysosome is absent in prokaryotic cells, but they are present in the eukaryotic cells. Now, RNA and protein synthesis takes place in the same compartment. But in the case of eukaryotic cells, RNA is synthesized in nucleolus and proteins in the cytosol or cytoplasm. Now, let us discuss about the cell organelles. This is a eukaryotic cell, and these are the cell organelles. Cell organelles are so called because they function like a different organ. They are all bounded by the membranes, separate membranes, and have got separate functions. Now this is the cell membrane or plasma membrane. Then this is mitochondria. This is lysosome. This is endosome, mitochondria. Now these are the Golgi apparatus, and this is endoplasmic reticulum. It is present surrounding the nucleus. Endoplasmic reticulum are having small, they are called rough endoplasmic reticulum when they have presence of ribosome on their membrane. And this is the nucleus, and this is nucleolus. Nucleus has several pores which are called nucleopores, and the genetic material are present in condensed form or chromatin in the nucleus. And this is nucleolus, which is rich in RNA synthesizing enzymes. Now, let us discuss all the organelles one by one. Let us discuss about the nucleus first of all. Nuclear envelope, double layer, nucleus is enveloped by a double layered membrane. Or this is the nuclear membrane. Actually, all the organelles are bound by the membranes which are double layered. So this is nuclear membrane. Inside is present the material which form the nucleus. And this membrane is having pores which are called nucleopores. Then chromatin or DNA is in the nucleus is coiled into a dense mass called chromatin. This is the dense mass which forms the chromatin. It stains darkly with certain dyes. So we can stain the chromatin or DNA with the help of the dyes. So we can locate them very easily under the microscopes. These are the chromatins. Chromatid. Now, nucleolus. This is nucleolus. A second dense mass closely associated with the inner nuclear envelope is called nucleolus. This is nucleolus. Now, nucleoplasm. Nucleoplasm of nucleus contains various enzymes such as DNA polymerase and RNA polymerase for mRNA and tRNA synthesis. So, this is the structure of a nucleus. This is nucleolus. This is chromatin, nuclear envelope or nuclear membrane, which is porous in nature. See, we can see the pores. And nucleus is surrounded by endoplasmic reticulum. This is the endoplasmic reticulum. And this endoplasmic reticulum is having rough surface or having ribosome on its surface. These are the ribosomes present in its surface. So they are known as rough endoplasmic reticulum. And we know that ribosome helps in protein synthesis. So some enzymes or something like that, proteins which are synthesized here in the endoplasmic reticulum. So just come to next. Let us discuss about the functions of the nucleus. One is DNA replication or the synthesis of its own replica, DNA's own replica, which is required during the cell division because cell has to divide into the same type of two cells. And RNA transcription. So the functions of the nucleus is one, DNA replication, another RNA transcription. The RNA transcription of DNA occurs in the nucleus. Transcription is the first step in the expression of the genetic information and is the major metabolic activity of the nucleus. So transcription is the first step of the protein synthesis. So DNA is first of all transcribed into RNA, and then the protein synthesis, it migrates into the cytoplasm where the protein synthesis takes place. The nucleolus is non-membranous and contains the various enzymes like RNA polymerase, RNase, ATPase and other enzyme, but no, it doesn't have DNA polymerase. That is, it cannot synthesize, the nucleolus cannot synthesize DNA. DNA polymerase is required for the synthesis of the DNA. Nucleolus is the site of synthesis of ribosomal RNA or rRNA. Nucleolus is also the major site where ribosomes subunits are assembled. Now, now the second organelle which we are going to discuss is mitochondria. It is also very important. Mitochondria is the powerhouse of cell. The number of mitochondria in a cell varies dramatically. Some algae contains only one mitochondria, whereas the protozoan contains 500,000. A mammalian liver, which is very active, it contains 800 to 2,500 mitochondria. Size. They vary greatly in size. A typical mammalian mitochondria has a diameter of 0.2 to 0.8 microns and a length of 0.5 to 1 micrometer. Shape. The shape of mitochondria is not static, it changes. Mitochondria, depending upon its functional stage, mitochondria assumes many different shapes under different metabolic conditions. Structure and functions of the mitochondria is that mitochondria is bounded by two concentric membranes that have markedly different properties and biological functions. So this is the structure of the mitochondria. This is the outer membrane. So you can see this, and this is the inner membrane. And inner membrane is folded, and these folds are called cristae. We know that in biological system, the folds are there to increase the surface area of that particular organ. So the mitochondria is a very active organelle. So the cristae or folds are present in it so as to increase the surface area of the mitochondria. And this is the inner matrix in which are present, this is mitochondrial DNA. Now this is intermembrane space, which is very much like the cytosol. Its composition is very much like the cytosol. Now even ribosomes are present here on the mitochondria. So mitochondria has an outer mitochondrial membrane, which I have just now shown. It is made up of lipid bilayer and proteins. Proteins are the present in the mitochondrial membrane are porins and enzymes. We know that all the enzymes are protein in nature. So the enzymes which are functioning in the mitochondrial function, they are present in this area, in this region. Now, inner mitochondrial membrane have cristae. That is, cristae are, just now I have discussed, are the folds which are present so as to increase the surface area of the inner membrane. And it is rich in proteins, then in lipid, and it is also impermeable. And cristae disappears when the mitochondria is activated. And then intermembrane space, which I have shown just now, acts like cytosol and have same composition like the cytosol. So mitochondrial matrix, the region enclosed by the inner membrane, is known as the mitochondrial matrix. Matrix also contains circular DNA, which I have shown just now in that figure of the mitochondria, has ribosomes and enzymes required for the biosynthesis of the proteins coded in the mitochondrial genome. Functions of the mitochondria. It is the powerhouse of the cell, that is, it helps in the synthesis of the ATP. So, generation of power in the cell, ATP from TCA and oxidative phosphorylation, which takes place in the mitochondria, and conversion of NADH and FAD to ATP to release them for contribution in the reaction again. Now, let us come to the structure of the endoplasmic reticulum. It is a membrane complex that are interconnected by separate organelles. They vary in shape, size, and amount. The endoplasmic reticulum extends from the cell membrane, coats of the nucleus surrounding the mitochondria, and appears to connect directly to the Golgi apparatus. These membranes and the aqueous channels they enclose are called cisternae. There are two types of endoplasmic reticulum. Rough surfaced endoplasmic reticulum, which is present near the nucleus, contains ribosome. Why they are called rough surface? Because they have presence of ribosome on their surface. And smooth surfaced endoplasmic reticulum, which have no ribosome. So if the endoplasmic reticulum is having the presence of ribosome on its surface, they are called rough endoplasmic reticulum, and they are known as smooth surface endoplasmic reticulum when there is absence of ribosome.